Okay, sir. You can start, sir.
Okay, sir. Yeah. So, sir, my voice is audible to you.
Yes, yes, very much.
So, dear all participants, Dr. Deshmukh sir, all eminent all eminent scientists who joined to the meeting, including the Dr. Prakash Halami and most respected Deshmukh Saab, and today's speaker, Dr. Subhadra Rathi. I all behalf from the Microbiology Society India and Gujarat Technological University Department is a school of applied science and technology. I welcome to all of you today's this webinar. So today's our eminent speaker is Dr. Subhadra Hati. So I'm giving a small introduction of Subhadra Hati. He having an excellent CV and excellent work he done in the area of the dairy microbiology. So at present, Dr. Hati is working as an assistant professor in the Department of Dairy Microbiology, SMC College of Dairy Science, Kamdhenu University, Anand, Gujarat, since 2012. Dr. Hati did Master of Doctorate from the National Dairy Research Institute, Karnal, in a dairy microbiology. Dr. Hati also did postdoc from the University of Queensland, Australia. He published more than 130 research papers in reputed journals, nationally and internationally, published more than 10 reviews paper, and edited 3 books. He He also handled externally funded projects from the DST, DBT, ICR, and Government of Gujarat as PI and co-PI. In the case of award, Dr. Rati is a recipient of numerous prestigious awards, including University Topper as a gold medalist in the undergraduate program, Teacher Award by the ICR and AAU, then NAS Young Scientist Award in 2019, then INSA Visiting Scientist Award by INSA New Delhi, and he is also recipient of Dr. Gauri Ganguly Memorial Award in 2019 by Indian Science Congress Association, Kolkata. He also awarded as the Best Innovator Award by the Microbiology Society India. He is also elected fellow member of National Academy of Sciences and associate fellow of NAST, WAST, and NADSI. He also received a BRICS Young Scientist Award in 2021 by DST. Recently, Dr. Hate delivered an invited talk at Obihiro University, Hokkaido in Japan as an international visiting academic. With this, I can surely tell you that the all audience, today's lecture is very, very important. Dr. Hati having a very wide experience and a wide exposure of the scientific communities and his domain. So now I request Dr. Hati to please deliver his talk. Dr. Hati, you can present your screen.
It is asking the host disable attendee screen sharing. So I should.
Yeah.
You may
Yes.
give me the permission or.
Yes. No, Dr. Deshmukh will give you the permission.
Please give me the permission.
Deshmukh, sir.
Because it is showing host disabled attendee screen sharing.
Yeah, Doctor, Miss, he is the host basically.
Yes, yes.
Yeah.
Deshmukh sir, please make me enable to share my slide.
Sir, I will call the Dr. Deshmukh. Just wait.
Yeah.
Sir, please give me, sir.
Sorry, sir. Within 1 minute. Within 1 minute. Yes, now you are permitted, sir.
Yeah. My slide is visible
Yes, sir. Your slide is visible. Please go ahead, sir.
and it is in the slide sharing mode, right?
Yeah, please make it full screen if possible.
It is not full screen now,
I'm not getting in that. Yeah, but now it is visible, sir.
but it is not in full screen.
No, but you can go ahead. It is completely visible to us. Perfect.
But my
Yeah,
what?
now it is visible, sir. Now it is. Please go ahead.
Okay, okay. So, good evening, all. First of all, thank you, Dr. Assis, for very nice introductions giving to me. And I am really thankful to President Microbiology Society of India, Deshmukh sir, for he actually requested me to deliver a lecture on this topic, technology and technological and health effects of buttermilk, because all of you know that who are in this society and who are in the different groups of the microbiology society that basically Deshmukh sir wants to make the buttermilk consumed daily, because there is a lot of health benefits, and it is our pride that we should maintain our good health as well as the nutrition. So one of the meeting he requested me, or he asked me to deliver a lecture. So I always I accepted that request and accepted that uh, uh, things that I will deliver, sir, but give me a 1 week time. So my two students and me both prepared this lecture. Okay, both the students are in this uh, virtually. They have also joined, so they also helped me to prepare this lecture. Even though we are working here on fermented milks, basically, even buttermilk or yogurt or dahi or other lassi, whatever it is, the fermented milks, we are working since 3-4 decades, because our institute is quite old institute, more than 60-65 years old institute, SMC College of Dairy Science. So I'll start with a very brief introduction about the buttermilk. Buttermilk referred to as the liquid leftover from the churning butter from cultured or fermented cream. Basically, you know that the drinks, name of the drinks, buttermilk, it is coming from the word butter. Okay, so when you prepare the butter, the byproduct coming out of the butter preparations, that is being used for fermentation, and then it will be used as a buttermilk. So that's why the name suggests itself it is a buttermilk. So it is coming from the butter itself. But nowadays, the commercially it is prepared in a different way. I will discuss later by one in the next subsequent slides. And in basically in western part of the country, uh, Chas, this is the traditional name of the buttermilk. It is very popular in Gujarat. Okay, everybody takes buttermilk in lunch or dinner. If you visit any of the restaurants for a dinner or lunch in Gujarat, if you don't ask them that you are not order buttermilk, then they will ask you, do you require buttermilk? So it is part of your lunch. Means it is very much close to have a good food. Then you must take the buttermilk. You see, when you go to the Western countries, because I visited 6-7 countries in Western countries, they basically takes the Coke in their food, basically in lunch or dinner. But here, because their food style is different, I will not say that they should not consume, but their food style is different. They may consume. But here, what kind of food we generally eat in dinner or lunch? The buttermilk is one kind of uh, helping food that will help you in digest, that will help to maintain your gut, and also it will give a bit more nutritious to the human beings. And this buttermilk is consumed all over the country in different names. It is called Matha in Hindi-speaking North India. It is called More in Tamil, Modu in Malayalam, and Majige in Kannada or Telugu, and Tak or Tak in Marathi, and Bhol in West Bengal. So different states have different names, but the thing is almost similar, but their processing parameters or processing method a little bit different. Buttermilk is a byproduct of the butter production, as I already told you. So the name itself comes from the butter. Is a yellow color liquid released from the churning of cream. When we prepare butter from cream, this byproduct generally released or generally generated from the cream, and it contains a lot of good nutrients like proteins, fat, oligosaccharides, phospholipids, glycolipids, and other constituents with having different biological activities. I will discuss different biological activities, what are the importance of the buttermilk in human health. And also buttermilk contains some amount of bioactive peptides, because during the fermentation of the buttermilk, these lactic acid bacteria, they produce different proteolytic enzymes, and that enzymes release the bioactive peptides in short fragments of the proteins, and it has different biofunctional attributes. Buttermilk is also a good source of calcium because it is made up of milk. You know, the milk is a very good source of calcium. So obviously it will transfer to the buttermilk and other nutrients like riboflavin, phosphorus, and potassium. It is also rich with proteins with a high nutritional values and functional properties that can enhance the quality of the food products. Its multifunctionality is utilized in improving the water holding capacity, texture, protection against the lipid oxidation in bakery, confectionery, sauce type of products. And the utilization of buttermilk extends to the production of functional beverages, cheese, ice cream, enhancing the quality of the final product. So basically, buttermilk can be consumed directly after fermentation by making a different, by adding some different spices or herbs. So this will be consumed as a beverage. Even though the dairy industry, they are adding this buttermilk for standardization of different product formulations. Okay, if they want to have more wherever there is a requirement of water, they are utilizing the buttermilk for making ice cream, for making cheese, or any other functional beverage. So they are basically utilizing the buttermilk not only as a beverage for making other products. Also, they are utilizing the buttermilk because they have a lot of fat and other nutrients. So it will mask off, and this is the huge amount of buttermilk is generated where there is a huge amount of butter is produced. I can take the example of Sumul Dairy in Gujarat. This is in Surat. So they produced a huge amount of butter. So daily they are producing lot of buttermilk. So not only they are making a beverage, they are also utilizing this buttermilk for different formulation of the products. So we know that the buttermilk, when we are preparing, we are fermenting it. Nowadays, Amul and other dairy industries, they are making the probiotic buttermilk. Most of the buttermilk now Amul is selling all over the country, they are using the probiotic bacteria. So the concept of the probiotic bacteria came in 20th century by the Russian Nobel Prize winner. He is also called father of modern immunology, Eli Mechnikov. He's a scientist at the Pasteur Institute and was first to conceptualize the probiotics. In 1907, Mechnikov proposed that the acid-producing bacteria in the fermented milk products could prevent fouling in the large intestine and, if consumed largely, lead to a longer and healthier life. So the probiotics concept basically given by him, and he established that if we take these bacteria in a good amount daily, we can have healthier intestine, healthier gut, and we live longer. In early 1930s in Japan, the name of the scientist is Minoru Shirota. He developed a fermented milk which is very popular all over the world called Yakult. And the bacteria which is used for making the Yakult is Lactobacillus casei shirota. And these bacteria is making wonders all over the world. The single bacteria can prevent different kinds of diseases. They are working in their lab in Tokyo, the Yakult lab, and these bacteria is very potentials, and Yakult is all very popular fermented milk beverage drink, probiotic drink, and people consume daily. The probiotic, basically the term probiotics given by the Lilly and Stilwell in 1956. And this is the history of the probiotics. Louis Pasteur in 1857, he basically says that there is a bacteria called lactobacillus, which have a very potential towards the health benefits. In 1878, Joseph Lister, he isolated first the lactic acid bacteria or lactobacillus mainly. Then 1899, Henry Tisser, he isolated the bifidobacteria, that is very potent bacteria. And 1905, Eli Mechnikov, which we discussed in the previous slides, he conceived the science basically of probiotics. And 1930, Minoru Shirota, he showed that how the probiotic product can be marketed all over the world for making or nourishing the human health. So this is the history to the application of the probiotics. Probiotic, the definition of the probiotic is given by different organizations, mainly the FAO and WHO. They said that it is a live microorganisms that, when administered in adequate amounts, confer health benefits on the host. But this IS, this is called basically Probiotic and Prebiotic Society. They said that live microorganisms which, when administered in adequate amounts, confer a health benefit on the host. So basically, it is almost similar, but only they have replaced that with which. This is ISAP definitions. So this is a basically what they want to say, that the microorganisms should be live and they should be in good amount. Their number should be in good amount, so that the host means the humans who are consuming the probiotic products, they get some kind of health benefits apart from the nutrition. Factors affecting the intestinal microecosystems. Basically, when we pass through different diseases, we take the antibiotics. If you have some microbial infections, or you are eating some processed foods where the fiber content is very less, and you are feeling or you are having some kind of diarrhea, chronic diarrhea, or you are under stress. So these kind of factors will generally affect your intestine. Intestine means your gut. So the probiotics have the role to restore your dysbiosis or your disbalanced gut and make you healthy. And probiotics, basically, it is a very old science is involved in probiotics. Somebody says that probiotic doesn't have any effect, but it is not like that. People are working since long, and I have given one data in 2000 up to 2018, around 4,900 papers already published on gut microbiota and probiotics. Okay, so people all over the world. Prakash Halami sir is here. He is also a very pioneer scientist in probiotic science and fermented foods. So all over the world, even in our country, people are working on probiotics, and these bacteria has. I'm not telling that this will work as a medicine, but definitely it will help your gut to make healthy. So imbalance of intestinal microflora results in poor nutritional response, reduce the efficacy of the medications, physiological dysfunctions, accelerated the aging process, even some kind of cancer and tumor proliferations, deficient in the immune response, susceptibility to infections, and physical discomfort. So these are the consequences if your gut is not healthy and you are having some kind of disbalance in your gut, means the bad bacteria number will be more than the good bacteria, then these kind of consequences will take place. So the probiotics is the solution for that. But when we select the probiotics, we should have to remember some kind of selection criteria. First is the safety, because you will consume it along with your foods, fermented foods. So safety is the very important part. You have to make sure that the bacteria doesn't cause any kind of harms to you. They are not containing any kind of virulent genes or pathogenicity, or they are not producing any kind of toxins in your systems. And the origin from where you are isolating the probiotics. Then functional aspects means techno-functional aspects, how they are producing acid, flavor, how they are growing in the milk medium or other food fermentations. Then how they are surviving. So when you will eat those probiotic products, they must uh, survive the acid tolerance in the stomach, or they should colonize in the intestine, and they must show some kind of antimicrobial effects. They give the immunity to the host, and they always maintain their genetic stability. They should not be participate with the pathogens for exchanging the and AMR genes or other genes through horizontal gene transfer or vertical gene transfer, basically horizontal gene transfer. And they must prevent the unhealthy organisms we call pathogens, and they should be stable during the processing of foods when you are making different kind of fermented products. Major prerequisite for the probiotic, it should be non-pathogenic, non-toxic, non-allergic. It should be capable of surviving and metabolizing in upper gastrointestinal tract secretion in the gut environment. And it always resistant. I told you that it should be resistant to low pH, organic acids, bile juice, saliva, and gastric acids. And it should be human in origin, genetically stable, and capable of remaining viable for long periods in the field conditions. And it should be able to modulate the immune response and provide resistance to disease through improved immunity or by the production of antimicrobial substance in the guts. And it also should have good adhesion and colonization to the human intestinal tract and influence on gut mucosal permeability. It is very important role for probiotics to maintain when you are eating the probiotics through fermented foods or through directly, because you can see the intestinal structure, how the intestines basically allow the probiotics or probiotic related metabolites to get entered into the circulation systems, because permeability is very important. Otherwise, the pathogens or bad bacteria, they can get entered in your circulations and they will make you harm and sick. It should be antagonistic against the carcinogenic and pathogenic organisms. It should possess the clinically proven health benefits. Maybe it could act against gastrointestinal disorder, diarrhea, Clostridium difficile colitis, antibiotic associated diarrhea, or acute infantile gastroenteritis. It should have a technologic properties for commercial viability, such as stability of desired characteristics during processing, storage, and transportations after making a final product. So this is the ICMR DBT guidelines followed in our country for calling any starter cultures, any lactic acid bacteria or other bacteria as a probiotics. It's a very important guidelines because you are doing some kind of health claim. So first is that the strain identifications, genus, species, or strain international collect. If you are collecting from some other culture collection center, then functional characterization through in vitro test or by through animal model, then safety assessment of that bacteria through in vitro or in animal model, or if you can able to do the phase 1 through human study, and efficacy assessment, it is called phase 2, you have to do clinically, and effectiveness assessment also through clinically. You have to prove that compare the probiotic with the standard treatments of a specific condition. So this is the guidelines available in the ICMR DBT. It is already published, the entire guideline. So you have to follow when you are calling that your culture is a probiotic cultures. And what is the status of probiotics today? The subject of intense medical research, the source of a multi-billion-dollar global industry, and National Institute of Health, they are funded for human microbiome project. Today, over 60% of the functional food products are directed towards the digestive health with probiotics, and probiotic science constantly evolving in different fields, and it is exploring widely. Now the question is why probiotics are being chosen through dairy matrix. Why the dairy matrix are very important to carry the probiotic bacteria. Because the dairy matrix are rich source of carbon and essential amino acids as a consequence of lactose hydrolyzing and proteolytic systems, which are being present in the lactic acid bacteria, and they can hydrolyze the casein. This is the major protein of the milk. Around 80% of total protein is casein. And milk has a good buffering capacity, also the fat content, which provide a suitable conditions for probiotics to better tolerate the harsh condition in the GI tract when it passes through the GI tract or the stomach. Some of the fermented milks like kefir, kumis, acidophilus milk, yogurt, they also give some kind of protection to the probiotic bacteria when it will be uh, carried by these of the fermented milk. So you can understand that your buttermilk is a good source of or good carrier of these probiotics organisms. So basically, dairy matrix are chosen or fermented milk products are chosen most to carry or to deliver the probiotic organisms. What are the effective factors on viability of probiotics? As we discussed, the intrinsic factors like pH, tolerance, dietary acidity, oxygen, water activity, presence of salt, buffering capacity, and some of the probiotics produce the bacteriocin or antimicrobial substances. They produce some kind of flavoring agents. Then processing parameters, which they have to tolerate like incubation temperature, heat treatment, cooling, storage condition, and packaging. Then microbial properties, the type of probiotic strain you are using for a particular product, interaction with the other microorganisms if you are making a symbiotic products, or the rate of inoculation you can optimize. So these are the different factors basically related to the viability of the probiotics. Then how you can add the probiotics by making a different fermented milks? First, you can add the probiotic microorganism together with the starter cultures. Most of the people do like that, because if I take an example of probiotic lassi made with Bifidobacterium bifida, okay, so what they do in a commercially, most of the dairy industries or fermented industry, they first make the curd with a yogurt culture, Streptococcus thermophilus and Lactobacillus bulgaricus. After making a curd, then when they are making it beverage out of the curd, they add this bifidobacteria, because bifidobacteria has some kind of, you know, they grow well, but they take some time because they are slow grower compared to the yogurt cultures. So that's why, and bifidobacteria is completely a strict anaerobic, and lactic acid bacteria are facultative anaerobic. So here you can understand that probiotic uh, bacteria can be used along with the starter cultures, because what is your purpose, what is the product basically it is, then you have to decide. Then addition of high viable count of the probiotic microorganisms of the fermentation stages. So at what stage you want to add the cultures so that you will get maximum count. Then incorporation of probiotic as a starter culture also. Just you can take the example of acidophilus milk. In the acidophilus milk, it is prepared with the lactobacillus acidophilus itself. It is a single culture. You can make the curd with lactobacillus acidophilus, because I worked on lactobacillus acidophilus in my master's program at NDRI, and I used only single culture, and that cultures have a very good antimicrobial effect. If you have a anti-diarrheal or any kind of food infections related problem, you can take the acidophilus. Lactobacillus acidophilus is wandering cultures for maintaining your digestion. Okay, so this is the single culture you can make the product out of this particular probiotics. Now I am coming back to the buttermilk, probiotic buttermilk. Basically, it is a the flow diagram I am sharing with you. It is followed in a very commercial dairy uh, who standardize the process. I have taken from them. They first take the standardized pasteurized milk. Standardized means how much fat will be there, how much SNF, solids not fat, because when you will make any kind of curd, dahi from milk, your SNF content is very important, solids not fat, because you know that how the curd is formed. Curd is formed from the protein, milk protein. So protein quality should be very good, and the lactose, because they will make you, lactose will be converted to lactic acid, it will lower down the pH of the milk, and then protein will get disturbed. Disturbed means they are pH, because proteins are pH sensitive. So when you standardize, or when you use any milk for making a curd or any fermented milk product, you should standardize the milk. You should know that how much protein should be there in the milk, how much lactose should be there. Fat is also important, because fat will give you some kind of flavor and mouthfeel. But protein and lactose is very much important for any kind of fermented milks. So that's why we require standardization. Then we have to give heat treatment, okay, at 90 degrees Celsius for 6 minutes to kill the native microflora of the milk, so that your bacteria will grow well. Unwanted bacteria will not come along with your own bacteria on starter cultures. Then we'll cool it down to 41 around if it is a thermophilic cultures. And generally, I have taken this flow diagram from a commercially available company. So they use a DVS culture, diet bed set cultures, and the trade name of that culture is ABY. Okay, it is basically trade name, and ABY contains three different cultures, uh, four different cultures. One is Bifidobacterium, another is the Streptococcus thermophilus and Lactobacillus bulgaricus, and another one is the Lactobacillus acidophilus. So here, Lactobacillus acidophilus and Bifidobacterium both are probiotics, but starter cultures are Thermophilus and Lactobacillus bulgaricus. So you can understand that starter culture is completely different from the probiotics. When you call it a probiotics, your culture must be passed through, which we discussed in the previous slides. So here they have used probiotics along with the starter cultures. So they first they basically want to make the dahi within 4 to 5 hours, maximum 6 hours. And these are the adjunct type cultures, Bifidobacterium and Lactobacillus acidophilus, they are probiotic cultures. So ABUI means A means acidophilus, B means bifidobacteria, Y means yogurt culture. Yogurt means Thermophilus and vulgaricus. So they have added as per their optimized amount. They have already decided by the Christian Hansen. It is the culture of the Christian Hansen. It is the Denmark-based company. Then we go for the incubation at that particular temperature for 4 to 6 hours. Then after making a card, we add salt and water, which we have, or the company or the industry who are making this kind of product, they have optimized it with the help of the sensory analysis, how the peoples like it. Then after adding salt and water, then they immediately chill the product around 10 degrees Celsius or below 10 degrees Celsius. Then they go for packing line for packaging that beverage in different SKUs, in different quantities, 200 ml, 500 ml, or 1 liter, what the pack is, or in PET bottle, or in even in the poly pack. Then they store it in a cold place, and when there is a requirement of dispatch, they send the product to the market. This study is being done in our college. We have prepared the buttermilk by utilizing the whey. Whey is a byproduct of paneer or cheese industry. So first, whey and double tone milk were heated to 90 degrees Celsius for 5 minutes to kill all living bacteria and cool to 37 degrees Celsius. Then the whey and milk were inoculated with Lactobacillus helveticus. It is our own cultures, and it is a clinically proven probiotic cultures. And we have used the starter culture from NDRI. This is NCDC 60, Lactobacillus lactis subspecies diacetyl lactis, at the rate of 2%. Then incubated at 37 degrees Celsius for 48 hours. It is a quite long incubation time. Then fermented whey and dahi were mixed into two different ratios, 60:40, 70:30, and control was prepared by using water in case of fermented whey in 70:30 ratios. So these are the results basically found or observed in the study. So there are 5 days storage life or shelf life. The titratable acidity, you can see that from day 1 up to 5 days, the acidity is being increased for 60:40, 0.68% lactic acid to 0.80 at 5 days. And in control, it is not so much high, up to 0.7. So because the storage temperature is around 4 degrees Celsius, 4 to 6 degree in between. Then also we have done the sensory analysis of these 3 different products. Okay, and up to, you can see that 5 days, the score generally does not go below 6, because we have measured in 9-point Hednick scale, and for different parameters of the sensory attributes like color and appearance, then body textures, then overall acceptability. So you can see up to 5 days, the score is around 6.85 to 7.6. So basically the rule is, when the sensory score goes below 6, then you have to stop the sensory analysis. Okay, so sensory attributes shows that the quality up to 5 days was okay. Then also we have checked the microbiological analysis of that product at different days, up to 5 days, and we did not find any kind of coliform counts and yeast and mold count. It was absent in 1 ml. And in lactic counts, you can see it is expressed in log CFU per gram, and it is almost maintained throughout the storage life, around 10.59 to 10. Very significant, non-significant reduction was observed, but almost it is at par with the storage days. Then again, they have also checked the biofunctional properties of that buttermilk, cultured buttermilk or fermented buttermilk. The ACE inhibitory activity, it is called anti-hypertensive activity, and you can see that ACE inhibitory activity for these 2 cultures, RT3 and RT4, where 60:40 and 70:30 ratio was maintained, is giving very good ACE inhibitory activity than the control, where only water and dahi is mixed. Then antimicrobial activity against Staphylococcus aureus, E. coli, and Salmonella typhi. You can see they have observed a little bit of antimicrobial activity of this product compared to the control. Then they have also checked the antioxidant activity. Again, you can see that 60:40 ratio is giving maximum antioxidant activity or free radical scavenging activity through ABTS method compared to the control. There is another buttermilk preparation by different cultures. There are 4 different variants are here. So they have taken the last flow diagram I was talking about with the milk, but here they have prepared the buttermilk from cream. So they have taken pasteurized cream and then they fermented the cream using different cultures. Okay, different cultures like aromatic cultures or aromatic plus bioprotective cultures, then probiotic culture, then probiotic plus bioprotective cultures. Then after cream ripening means after fermentations, they churn it, then they make the, after churning you will get the butter grains, and the byproduct of the butter grains is the buttermilk. So they transfer this buttermilk to the another line. This is called cultured buttermilk, and then they follow the other pathways to make the butter sample. So basically here you can see the cream is being used as a raw ingredients, and from cream they have prepared the butter samples with the different culture combinations with the different batches, and then the byproduct of that butter is a cultured buttermilk. They are packaging and then they use it. So it is the basic uh, preparation of the buttermilk from the cream or butter making process. And they have analyzed the physiochemical properties of the buttermilk samples. They have checked the dry matter, fat, acids, pH, titratable acidity in terms of lactic acids. And most of the cases they have, because they have used different types of cultures, aromatic cultures, then aromatic plus this one, bioprotective cultures, then probiotics, then probiotic and bioprotective cultures. So there are different cultures they have used, and it is not in case of ash and fat. Fat, I have some kind of significant difference, but pH, there is no significant difference. Even titratable acidity, only the probiotic culture is giving more lactic acid than the other bacteria. Viscosity, you can see there's the probiotic plus bioprotective cultures giving more viscosity. I don't know how it is more, but not so significant, what I can understand from the data. One study I got it from the very recent study they have done on sheep milk. Okay, they have used the sheep milk for making a butter, and from that butter they have got the buttermilk, and means the cultured buttermilk, and they have said they have observed the bacterial loads in the butter as well as the buttermilk, fermented buttermilk. So you can see there in the, this is a butter and this is the butter milk which is being produced from this butter. It is a lactobacilli count in butter and it is the lactobacilli count in the buttermilk. Basically buttermilk, they have used different cultures, aromatic, kefir and probiotics. And here also they have used different cultures for making butter, means this is the fermented butter. And then lactococci culture is also used here for making, this is different kinds of butter, sweet butter, aromatic butter, kefir butter and probiotics. And they have done this storage life up to 90 days and for buttermilk up to 28 days. So you can see that the probiotics are giving more counts in the buttermilk compared to the butter. Uh, at first days, even in the 28 days, the count is around log, more than log 8. But in butter, it is not going more than 8. Okay. Similar for the lactococci count also, you can see the probiotics are giving very good amount of counts. It is around 11.5 log CFU per ml in first day, as well as in the 28 days it is more than 8.2, 8.3 log CFU. But in case of butter, you can see the count is around 6.5 or 6.7. So here you can see that the buttermilk is a good carrier of the uh, any lactic acid bacteria which are being used for fermentation of the buttermilk. And I already discussed what are the benefits of the probiotics, what are the benefits of the lactic acid bacteria. Apart from the nutrition of the buttermilk, then health benefits. What are the health benefits of the buttermilk? People with a lactose intolerance, they may not be able to digest the lactose. Whenever they will consume the milk, they may have the abdominal pain, bloating, diarrhea and flatulence. But they can take the buttermilk, because in the buttermilk, the microflora, the lactic acid bacteria, the good bacteria, they will hydrolyze the lactose and it will be converted to glucose and galactose, and they may not have this kind of problems. So the metabolic activity of the microbes convert the lactose into lactic acids, and which is easy to digest. Additionally, buttermilk also plays an assistive role in the removal of undesired stomach acids by creating a layer on the lining of the stomach and moving the acid through esophagus. So this kind of fermented beverage are very good for our health. Then hypocholesteromic activity. Hypocholesteromic actions of the buttermilk was studied using different methodologies that demonstrated a reduction in the total cholesterol and other variables with the consumption of the dairy derivatives in the short term, because they contains lot of phospholipids in the buttermilk, and it has a very good role on the hypocholesteromic effect, and that the effect occurs through an intervention in the pathway of cholesterol synthesis as well as in the intestinal absorption of cholesterol and lowering their blood levels. Means their level will get reduced at the circulations. It also has been suggested that buttermilk intake can reduce the cholesterol levels by inhibiting the assimilation of cholesterol in the intestinal tract also. Then anti-cancer effect. Some authors stated that anti-proliferative effects of several isolated fractions of buttermilk obtained from food and non-food solvents on some type of human cancerous cells. This study was done on cancer cell line. The fractions and rich in phospho and sphingolipids have a strong anti-proliferative actions on colon and ovary cancer cells. And these observations allow to hypothesize the use of these phospho and sphingolipids as a functional food additives. Further, it has been observed that consumption of cultured buttermilk can decrease the chances of bladder cancer. So these are the studies people have done. I can discuss about the anti-proliferative activity assay. It is an in vitro assay. It is based on the methods given by the Monks et al. And it was conducted to assess the effects of various samples on human cancer cell lines, Hackard cells, and cells were cultured in RPMI 1640 medium with a 5% FBS and antibiotics maintained at 37 degrees Celsius in a humidified atmosphere with 5% CO2 and used below passage 20. After 24 hours of incubation, cells were treated with the sample solution prepared by dissolving 5 mg each sample in DMSO, followed by dilution to different concentrations. And dexorubicin served as a positive control, and post 48-hour treatment, cells were fixed with a trichloric acetic acid stained with a sulfohordamine B, and the absorbance was measured at 540 nanometer to determine the cell proliferation. Cell growth inhibition percentage were calculated and monitored, and non-linear regression analysis was monitored to determine the concentration required for total growth inhibition and 50% growth, means GI50 values. So these are the 9 different cell lines was used to assess the anti-proliferative activity of the buttermilk. Okay, so there are 3 samples of the whole buttermilk powder and their lipid extract with a food grade or non-food grade solvents are prepared. So 3 samples they have prepared to check the anti-proliferation assay by using these 9 different cancer cell lines, human cancer cell lines. And doxorubicin at a different concentration was used as a positive control on cell death. The whole buttermilk powder sample, which contained proteins and lipids in its composition, did not show anti-proliferative activity in any of the 9 human cancer cell lines, but only the buttermilk fat extract obtained using food grade solvents showed anti-proliferative activity against K5G562 cell line. This is a leukemia cell line with a proliferation inhibition higher than 50%. So the buttermilk fat extracted by using the food grade solvent, they are showing anti-proliferative activity, and more than 50% results was observed. But cancer cell line proliferation was not inhibited by the buttermilk fat extract through the NFG non-food grade solvents. Although a higher activity of non-food grade buttermilk could be expected owing to the higher content of PLS1 in comparison with food grade buttermilk. So here food grade buttermilk showing the good results by inhibiting the cancer cell lines activity. Then people have made the buttermilk by supplementing the vitamin C in different concentration, 100 milligram per ml or 200, 300 and 400 milligram per 100 ml. And they have observed different tests. You can see here, they observe protein, fat, lactose assay, and there is no significant difference they have observed. And these vitamin C content, they did not interfere with the sensory attributes of the buttermilk. It doesn't affect the storage life of the product, and also it does not have any kind of compositional attributes of the buttermilk. So we can supplement vitamin C in the buttermilk wherever we have required. So you can understand, or you can imagine that the vitamins can be fortified in the buttermilk if it is compatible with the buttermilk matrix. Then one clinical study, I got it from some of the papers. They have done the effect of the buttermilk, or basically the cholesterol level, plasma lipids, and surrogate markers of cholesterol homeostasis in men and women. This study was carried out as a double-blind, randomized placebo-controlled crossover study, according to which the participants were subjected to 2 consecutive treatments of 4 weeks, but total duration is around 8 weeks. Okay, so the buttermilk powder was purchased from this company. This is the Westland Milk Products of New Zealand, and was fully characterized and formulated in ready-to-use pouches. So each pouch contained around 22.5 gram of formulated buttermilk or placebo that subjects had to mix with 250 ml of the water using a shaker provided by the study center. Participants were asked to consume 2 pouches every day just before breakfast and dinner, for a total consumption of 45 gram per day of the buttermilk or placebo, which corresponds to 5% to 10% of their daily energy intake based on a 2,500 kilocalorie per day diet. Participants also received specific instruction on how to introduce the test formulation in their diet by substituting specific food items. Subjects were also asked to refrain and to maintain their intake of dairy product to a maximum of 2 servings per day. This is the criteria of selecting the human subjects. So first they have recruited 116 people through telephonic screening, and from there they have selected 82 which are eligible for tests, and from 82 they have taken the attendance of who came to their place for participation in the study, around 77, and when they physically checked all these parameters, they have included only 42, and 40 peoples out of 40, 42, 2% did not come. So finally they have a 40 clinical subjects. But when they have started the work or started the study, they have found that some of them did not means acceptable because they have some kind of health issues on the time it is started. So 34 finally they have selected for this study. So total clinical subjects are 34, and they have checked. Then they have given these probiotic buttermilk, not probiotic, buttermilk given to them in a 2 servings per day. Okay, they have, I already discussed that for 8 weeks they have given, and they have monitored their health and other things. And after that they have checked their blood profile, mainly the lipid profile. So you can see by comparing with the control placebo with the buttermilk, uh, you can see that the total cholesterol in the placebo it is 5.92 millimolar per liter, and it is buttermilk it is 5.74. Then LDL cholesterol, this is the bad cholesterol, you can see it is 3.71 in the placebo, but it is 3.59 in the buttermilk who have consumed the buttermilk. Then apolipoprotein B100, it is 114, but the persons who are consuming the buttermilk it is 117 milligram per DL. Then good cholesterol HDL, it is 1.62, it is almost 1.62 for both. Then C/HDL ratio, it is 3.91 for control, but it is 3.77 for the buttermilk. So you can see that mostly the triglyceride level it is 1.3, but in buttermilk it is 1.6. Even in the apolipoprotein B48, it is 6.6 for the control, and it is 5.80 for the buttermilk. Even the beta citosterol also 0.79. In case of buttermilk, it is 0.76. So in clinically also, I am not telling that very significantly they have observed the result, but it has some impact on the cholesterol or lipid profile of these men and women they have recruited, and they have studied the effect of the buttermilk. So I'm almost in the end of this presentation or sessions. These are the different probiotic buttermilk or normal fermented buttermilks are available all over the country. Different companies, they are big dairy giants, they are selling lot of buttermilk or chas or probiotic buttermilk all over the country. People are consuming it. In summer, we know the consumption of buttermilk is very high to maintain your health, and also it is a thirst quenching beverage drink, and we have discussed that it has a lot of health benefits to consume daily with your lunch or with the dinner, or even if you feel that you are need some water or you need some kind of thirsting beverages, you can prefer this buttermilk. And I am really thankful to the Microbiology Society ki they are taking such kind of initiatives to make the buttermilk, to promote the buttermilk so that people consume daily. So now I am concluding the my talk here. Fermented probiotic buttermilk is a nutritious and it is a functional dairy product that offers numerous health benefits if it is probiotic cultures or you have add some kind of spices or herbs. People have added or people have developed some kind of novel bio buttermilk. I know they have made with different kinds of herbs and spices as well as the probiotics, and really it's a thirst quenching, and as well as it has a good kind of antioxidant or antimicrobial effect. Some of the probiotic buttermilk has a peptides, and they have anti-inflammatory, antimicrobial, anti-hypertensive properties. Fermented probiotic buttermilk is a viable addition to a healthy diet, and its potential health benefits make it exciting area of research and development in the field of functional foods. Further research is needed to fully explore the potential health benefits of fermented probiotic buttermilk and to develop new products with the enhanced nutritional and functional properties. So I'm really thankful to the Microbiology Society of India and uh, the GTU, Gujarat Technical University, because the Dr. Ashish, who is the convener of this today's webinar, he takes a lot of interest to organize this talk, and I am really thankful to the Microbiology Society that they have taken a very good initiative to popularize the beverage, basically the probiotic fermented buttermilk, which is not only healthy, it also gives some kinds of nutritions, and it is a very thirst quenching drink. So thank you all for listening patiently. Over to you, Dr. Ashish.
Yeah. Thank you, Dr. Hati, for delivering the extraordinary talk, I can say, because you highlight the all the things which is covered by the most popularly in India. It is a buttermilk and charge, right? And we are using it is from the ancient period, but you cover the entire scientific mechanism, and I can say the validation of the charge. Now the people are getting that knowledge and they are taking this as a probiotics, but as an instant time we are using that, but we don't know about the scientific reason. So now I open this forum for the question and discussion. So I request to all participants so they can actively participate in a discussion, or if you have any query, you kindly ask to the Dr. Hati, please.
I think Halamis have raised hand.
Yes, sir. Yes, Halami, sir, please.
Thank you, Ashish. Of course, big congrats to Dr. Subardo for giving such a illustrious talk covering why buttermilk is known as a buttermilk,
Yeah.
till
Yes.
the preclinical and clinical trial and the different products, range of products are available in the market. A very inspiring lecture. Of course, you worked with the NDRI, so the topic of your interest from your postgraduate time. Just a few questions which actually I came across the time MBSI has actually initiated this movement along across the country. You know that buttermilk we should drink. You know, so first one is of course a week back I was in Delhi and then I came across with Mother's Dairy's buttermilk where Bifidobacteria was incorporated, and I was unable to get whether they added the starter culture for that or what. So that is what I wanted to know. And then they claim some count of the Bifidobacteria, whether Bifidobacteria really can survive during the product preparation and during the viability. So this is my first question. Then I will, once you can tell, you put a highlight on this, then I will go to the second question perhaps.
Sir, actually, what you understand, I could know that you are asking that why bifidobacteria is not being used for preparation of the product, or you are asking that
No,
bifidobacteria.
bifidobacteria are incorporated in a buttermilk prepared by the mother's dairy in Delhi. Okay.
Yeah.
So
They
how
are
we are sure that bifidobacteria will really survive, and whether they, while the preparation of the buttermilk, whether they used any other adjuvant culture or a starter culture for
Yes.
curdling purpose.
Basically, sir,
Ah,
because I also worked in Mother Dairy Delhi in 2008, 2009. On that time, we
okay.
were preparing the buttermilk as well as some probiotic drinks called NutriFit. And we used
Mm-hmm.
the bifidobacteria that is called BB12, which is taken from Christian Hansen. It is called Bifidobacteria
Exactly.
animalis. Okay. But
Exactly.
the thing is that
Exactly.
we never add the bifidobacteria for making curd. We make the
Mm.
curd with the starter cultures, basically yogurt cultures. Within 4 hours, we got the
Yes.
curd. And then we
Yes.
break the curd with water and standardize at a particular SNF content. And when it
Yes.
is cooling down to around 4 to 6 degrees Celsius, when it is going to the packaging line, there is another tank
Yes.
where we
Yes.
add the bifidobacteria immediately and we pack it. Because you know that
Mm-hmm.
bifidobacteria is a strict anaerobes. If
Yes.
some amount of oxygen is there, it will not survive. Even
Mm.
though when we check the final counts of bifidobacteria, it is very difficult to get it because you know the
Mm.
bifidobacteria, if you do the incubate, your incubator should be a CO2 incubator. Even when you do the plating, there are
Exactly.
some chances that the oxygen will get incorporated.
Exactly.
And the media where basically we use the MRS, but we have to add some antibiotics so that other flora will not grow well because starter cultures
Exactly.
are there. So it is really difficult for bifidobacteria because bifidobacteria are excellent probiotics. There is no questions about it. But to make it survive, to deliver it into the site, it is very difficult. But Mother Dairy and even I can tell you the ABY. Na, I have given one example.
Yes. Yes. Yes.
Even Amul is
Yes.
selling buttermilk with ABY, probiotic dahi
Yes.
with ABY. ABY means bifidobacteria B, and acidophilus.
Exactly.
But what they are
Exactly.
doing, the FSSI has made some kind of relaxation. I
Mm.
can say they said the 10 to the power 6 counts per serving, not per gram. It is very
Mm-hmm.
difficult to reach nowadays. But not only
Mm.
the counts, we should check the
Mm.
activity also because during processing, count is one thing, yes, it is required. But are they
Exactly.
really give you this kind of, they should be physiological active enough to pass
Exactly.
these different harsh conditions of our GI tract. So again, I will suggest that the industries that not only you should take the counts, you have to also check some kind of in vitro probiotic tests with your product, means the cultures.
Mm.
Okay,
Yes.
and then only it will be claimed well. Yes, sir.
The second question is with relation to the health benefit, what people are actually claiming,
Yeah.
right? Where because when we make the buttermilk, we are actually basically diluting the culture,
Yes.
right? We are
Yes.
diluting the overall content, you know.
Yes.
So with for thirst purpose, for nutrition, it is all right, that lower fat content,
Yeah.
right? But with reference to the health benefits, when we see how much one should consume, how long one has to consume, is the
Yeah.
ultimate question, which the most part of your presentation was there about this probiotics and health benefits. So
Yeah.
when we talk about the buttermilk as a diluted product, right? Because from the curd we make 1:4 or 1:5. That means we are actually diluting everything,
Yeah.
right?
Yeah. True.
But we can't just be drinking liters of a buttermilk and telling I want to see the health benefits. So with your own experience, how long and how much we should consume, where we can actually extract that we get a good health benefit.
Sir, in concerned about the buttermilk as
Mm-hmm.
making or diluting all these things, not only bacteria, even the nutrition, also the proteins or other macronutrients. But
Okay.
the health claim, what you are asking that about the particular buttermilk, it is very difficult to
Ah,
health claim as a buttermilk. But as a culture, it can
exactly.
be done. But exact product, I have some doubt because I have given
Exactly.
one clinical study. They have basically they have mentioned that this amount of, but they are not probiotics. They did not claim for
Ah,
probiotics.
ah,
They have only
yes,
overall product
yes,
quality
yes.
they have checked. But for probiotic as a health claim for buttermilk, it will be very difficult. because I have checked the buttermilk which is selling by some of the companies. I don't want to take their names. Count to achieve the count is also difficult because they are diluting in such a way for maximizing their profit. Okay. Even the organic acids or short chain fatty acid I have analyzed in my lab. Okay. It is very less than the dahi in case of buttermilk. So for health claim, really it is difficult.
Exactly.
Think of the what you are asking about, that how much we should consume and
Yes.
at what period, means the duration of the particular buttermilk which are having health claim. Because Amul is selling probiotic buttermilk. They have written, I have shown you this slide. They are selling because
Yes, yes.
they said, they are not saying that any health claim. They are only saying it is a probiotic buttermilk.
Exactly.
Yes.
Just a last question. I hope Ashish has the time for me. I feel some other participants also have raised their hands. Last question, actually, we have a product, Bifidocard, and I want to convert that one as a buttermilk, buttermilk. In fact, I put this one in a group also. So, what is your comment, suggestion? In fact, on the 11th, coming 11th, we have one here, a industry connect workshop on probiotic, postbiotic, and fermented food. And in fact, Bifidocard technology has been already transferred to an industry. Right now, they are not making, but then I want to actually promote that somehow. So, my question is that Bifidocard is prepared exclusively using a single bifidobacterial culture. Okay. And then as you told that to make a different taste, flavor, you can add some ingredients. So with reference to this, if I have to use this,
you can prepare, sir, but the thing is that you have to make a card and then you can make the buttermilk. You can go for some spices, add some spices.
Exactly.
Yeah, salt and a bit salt, not more, but a little salt on sensory analysis.
Exactly.
You can set to it because the bifidobacteria, if they are making good card, then it will be very excellent buttermilk.
Exactly. And then we can really deliver a bifidobacteria in a quite a good number.
Yes.
So this is what the overall approach is, because lactic acid bacteria otherwise also we get through one or other product. Bifidobacteria, ingestion of this bifidobacteria is very limited through our diet.
Yes.
Right. So from my side, timing it is okay. Thank you very much, and once again congratulations for a nice presentation.
Dr. Mosmi.
Yeah.
Thank you, sir. Thank you. Dr. Mosmi.
Namaskar. It was an excellent talk, Dr. Subroto. I really liked the way you explained. I have one question. Like most of the children these days, they don't want milk. So for them, buttermilk would be a wonderful substitute. But there are so many children who also suffer from lactose intolerance. So for them, digestion or elevated symptomology, what would be there with this lactose tolerance in case of buttermilk?
Dr. Ashish, can you mute that number 813? It is disturbing.
The number having the 81, please, will you off the mic, please?
81066. Please mute yourself, or if you have a questions, we will take you in later on. So, Dr. Mosumi, you are concerned about the lactose intolerance for children, right?
Yeah.
But I already explained in my slide that fermented milk, not exactly the 100% lactose will be converted to the lactic acids, but we have estimated after fermenting milk in basically the curd, dahi, because in milk it is around 4.5 gram, 4 to 5 gram in 100 ml milk, lactose is there. But after fermentation, around 30 to 40% lactose will be converted, not 100%, okay, not 100%. But nowadays, what Amul is doing, they are utilizing the lactose hydrolyzed milk for fermentations. They are making the milk lactose hydrolyzed. It is available in the market. You can take that milk and you can add the cultures, and it is completely lactose free dahi. That is one option. And
Okay.
another option is that the buttermilk, as Prakash ji, Dr. Halami sir, we are talking about that when we are making a buttermilk out of dahi, it is getting diluted. Okay.
Yeah.
So your lactose, if some amount of the lactose around 40% or 50% is there, it will get diluted again. And
Yeah.
30 to 40% is already converted from lactose to lactic acids by the fermentations. So
Yes.
your buttermilk is a good choice for the children who having some kind of lactose intolerance problems, or any kind of fermented milk is also good choice for them.
Yeah, so what we can speak about for all Indian children with this notion of drink buttermilk and be healthy, that it contains very least lactose
Yeah,
intolerance. Yeah,
yeah.
so this is what I would
Good
like to speak to my
option.
students. And
Yeah,
just
good.
the second question, if time permits, that is, there are certain potential allergens or contaminants in the buttermilk also. Now, should we also highlight that?
Allergens, you see, allergens coming, buttermilk does not cause or produce any allergens. Basically, the allergens coming from the milk itself.
Ah,
The milk protein, it is basically called beta lactoglobulin. This is called milk allergen.
yes.
Okay. But in fermentation, again, I'm telling you, during the fermentation, the lactic acid bacteria has a very good proteolytic systems, because I am
Okay.
working on peptide production since 2014.
Hmm.
So these lactic acid bacteria, they have a very good proteolytic systems. Basically, these bifidobacteria or these Lactobacillus helveticus, when we make the product out of these cultures, they basically break down the beta lactoglobulin. So the
Okay.
fermented milk, again, it will help you to prevent the milk allergens. It is a good source for those who are having the milk allergens. They can take the fermented milk, because basically the milk allergen protein is the beta lactoglobulin, and the
Yes.
bacteria will hydrolyze this beta lactoglobulin, and they will disturb their structure, so it will not cause milk allergen directly. So again, your fermented milk is a good option for them, and buttermilk is itself, it's a good option again.
Just one last question. Most of the there are different brands, as you also showed.
Yeah.
They claim different shelf lives. So,
Yeah.
what is the basic reason of the different shelf life? Is it the bacteria that they are using as their base culture system? Is it due to that, or what do you think as a microbiologist should be the basic reason?
See, the shelf life basically we generally do in whenever we make any product, we do the sensory analysis. And apart from sensory analysis, we do the microbiological analysis and physicochemical analysis. But main important part is the sensory. Okay. So the product which you are telling that some of them are 10 days or 15 days or 28 days.
Yeah.
Okay. So the peoples, if they like up to 10 days and it is safe means the microbiological quality like coliform counts, eastern mold count, okay, and lactobacillus count, which is a beneficial bacteria, their count is maintained throughout the storage life, we consider that period. And if you are adding some kinds of other ingredients like herbs or spices, or because lactic acid bacteria, they will enhance your shelf life. You see, your milk, pasteurized milk, has only 2 days shelf life, 48 hours. But when it will convert it to the dahi or any fermented milk, their shelf lives going more than 10 days. So basically, fermentation concept and fermentation basically do the enhancing of the shelf life. It's a value addition for enhancing the shelf life. We call it a ESL, extended shelf life. So lactic acid bacteria is giving the good amount of shelf life. But the problem is if you are adding some contaminants along with this bacteria, or your packaging system is not properly maintained, or you are adding some herbs and spices which are not properly heat treated, or giving some kind of heat treatment so that they should not carry any native flora which cause the problems. So that's why the shelf life will be different depending on their processing treatment. We cannot say that every product have the similar kind of shelf life. Shelf life is depending on your ingredients in your processing parameters. And again, I am telling you that most important part to check the shelf life is the sensory analysis. How your consumers liking it. In a different scale, you can use it. So that's why the shelf life. But on an average, fermented milk or fermented beverage, it has 10 to 15 days shelf life when you are storing these products at refrigerated temperature around 4 to 6 degrees Celsius.
Thank you so much. Thank you so much.
Thank you.
Now,
Thank
Ashish
you.
sir,
Yes, sir.
I request you to conclude now.
Okay, but yes, sir. Sorry, but Dr. Hatia, I have one question, but I will ask you directly. Okay, no
Yes.
issues, right?
Okay.
No, you can
So,
ask. You can
thank-
ask last question.
Okay, so many agriculture scientists or so many agriculture farmers use this buttermilk for the protection of or the cure of their some plant diseases, right? So, any idea what is the reason behind this?
Main part is that this kind of questions already asked because in last 6 to 8 I was in Patna for a dairy industry conference,
Yeah.
and one fellow from agriculture background, he was asking that how these lactic acid bacteria can help us in plant pathogens. Okay, they are asking. So I did not work on it, but what I can understand by discussing with the agriculture microbiologist that our lactic acid bacteria produce some kind of organic acids. Okay,
Right.
and some kind of mostly the organic acid and some kind of antimicrobial substances like bacteriocins, okay, some hydrogen peroxides. So these are the antimicrobial substances directly affect the pathogens or pathogenic microbes. For it inhibits their growth.
Okay.
So the organic acid like lactic acid, acetic acid, butyric acids or propionic acids or other citric acids, so they produce it,
Yeah.
and they make the medium very acidic by producing organic acids, as well as it also produce the bacteriocins like antimicrobial substances.
Yeah.
So that's why maybe that is the reason, but
Yeah.
definitely we have to study on it.
Yeah. Thank you. Thank you very much. And here I'm giving the big thanks to Dr. Hati and Dr. Deshmukh, who is helping us for this arranging such a wonderful webinar. And Dr. Hati, I can say to you that more than 18 participants joined this program, and there are wonderful discussion held here with the Dr. Prakash Alami, the Dr. Mosmi, and it's a wonderful discussion. And this is the outcome of such a kind of the webinar. So I am from the GTU and from the Microbial Society and the president of the Microbial Society, Dr. Deshmukh sir. I say thank you very much. And now I hereby say that it is a conclusion of our seminar. So you may leave now. Thank you very much, Dr. Hati. Halami sir, thank you very much. Deshmukh sir, thank you very much.
Thank you. Thank you very much.
Yes, 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 yes.
Thank you, Deshmukh sir.
One more thing.
Huh, huh.
Yes, sir.
Today's lecture is recorded,
Yeah,
Yeah.
and tomorrow it will be circulated on YouTube.
great, sir, great.
Wonderful.
So it will be available for others also, those who have missed this lecture.
Yes,
Sure,
Yeah.
sir. Sure. Thank you.
Okay.
Thank you, Deshmukh sir.
Thank you.
Thank you. Thank you. Thank you.
Thank you, everyone.
Thank you, everyone. Good night.
Good night.